It's 12:10. Time for us to get started. It's a pleasure to be able to introduce our seminar speaker for today, Herman Nesselink from the Netherlands. I think many of you have probably written or read a little bit about what has been written about Herman. He got his degree working with Joop van Lenteren at Wageningen University. And many of you may know of Joop, but basically Joop van Lenteren really advanced biological control in greenhouses around the world. And he had a tremendous stable of students that now are again around the world still advancing biological control. Herman is one of them. After his degree, he worked a little bit with a plant diagnostic firm in the Netherlands, both insects and nematodes. Kind of got bored of doing that, and they moved into a research position at Nalvik, which is an experiment station very famous for developing biological control strategies for vegetable crops in the Netherlands and around the world. And people that know anything about biocontrol in greenhouses, the name Nalvik basically comes to mind because of the tremendous advances that have been made there. There have been some major changes in the Netherlands over the years. There used to be a research station at Aalsmeer, and then one at Nalvik. Aalsmeer did floriculture, Nalvik did vegetables. They've kind of combined them. Herman has sort of moved into a new position where he is, a new location, I guess, basically, where he is senior research scientist, and he does biological control, or practical biological control for the greenhouse industry, both vegetables and ornamentals in the Netherlands. He's a well-published researcher, and again, the focus is sort of understanding natural enemy biology and ecology with the idea of making use of these natural enemies in an applied context. And he works across a broad range of different systems, and I think you'll see some of that in his presentation today. So,
yes, thank you very much, Mike, also for this invitation. It's really wonderful to be here. I hope everyone can hear me very well. Is it okay? All right. So today I would like to tell something about the role of generalist predators in biological pest control. Because in the past, I think we used to focus a bit more on the specialist natural enemies, and it has shifted a bit more toward the use of generalist predators.
Move that up a little higher.
Yeah.
Let's see if you can.
Is this better?
Sorry.
Not really.
Not really. No.
And if I speak loud, is that better?
That's better.
Yeah. Try to speak loud. Not to shout. All right. So today I would like to tell something about these types of natural enemies, generalist predators in biocontrol. And my name is Herman Nesselink. So first of all, I work at Wageningen University and Research Center. It's a combination of applied research stations and the university. I'm in the plant science group here.
Excuse me one more time. Let me just see that. Let me replace the battery. Speak up. Yeah, sorry.
Yeah. Okay, so I'm working in the plant science group. Something about agriculture in the Netherlands. Horticulture is really important. I'm working in the greenhouse sector, but it's an important industry. About 70% of the production value is caused by greenhouse industry. Well, this is the Netherlands. We are a very small country in Europe. Close to the coast, but we have about 10,000 hectares of greenhouse industry, and it's mainly located in this area. It's called the Westland, and also our research station is in this area where all the industry is. It's about 50% floriculture and 50% vegetables, and there are also some other areas where they produce greenhouse industry but also here it was in the past more floriculture, and here more vegetables. So these are the important areas. My research station is located in Blijswijk, and part of our group is working in Wageningen. And our mission is to initiate and stimulate innovations for a sustainable greenhouse sector. It's both strategic and applied research, and we have about 80 researchers all working in this greenhouse industry. In Blijswijk, it's a location where I'm working. We have about 19 greenhouse compartments where we do all kinds of experiments, crop protection laboratories, flavor, taste facilities, and we also have an innovation and demonstration center for greenhouse as energy source. So there's a lot of research going on in energy saving in greenhouses, and they want to switch from energy consuming industry to energy producing industry. So that's quite challenging. There's a lot going on, but I will not tell something about that today. It's about IPM, Biological Control. Our funding, it's mainly based, it's project-based funding. A lot of funding comes still from the government, Ministry of Economic Affairs. It's about 50%. But we are a privatized institute. And a lot of funding is coming from the Horticulture Produce Board, about 30%, and some other funding, European Community, private companies. Well, something about the greenhouses. It's mainly, well, high-tech greenhouses, glass houses we have, not so much tunnels. And they appear very sterile. If you look at this tomato production, it's all in hydroponics, not in a soil. And it looks really clean, and it is also clean. And this is an example of a nice grower who's producing sweet pepper with artificial light, so it looks really nice. But although the greenhouses are really closed and it's high-tech, somehow always pests appear, and we have mainly the common pests you probably also have, or I'm sure you also have the whiteflies, and we have thrips coming in, one of the big problems, both in vegetables and ornamentals. We have aphids that appear, and we have spider mites that blow into your crop somehow. And yeah, just for those who are not so familiar with these pests, this is thrips, western flower thrips. We call it the Californian thrips, so it came from California somehow in the past. That's the story. Yeah, but it's a huge problem in many crops. It also transmitted viruses. We have whiteflies, greenhouse whitefly mainly, also Bemisia, but this is the most important whitefly. So this is a lot of honeydew excretion, a lot of problems. Spider mites, leaf miners, we have a lot of aphids. Yeah, a big problem in pepper is the green peach aphid, and the green peach aphid had also red phenotypes, biotypes, and they go immediately to the flowers. So what happens is that the flowers drop, you lose production, they excrete a lot of honeydew. So this is a big problem in the organic pepper industry. It's very hard to control. Another important aphid species are Locotra solani, the foxglove aphid. You see these strange symptoms on the leaves, a strong response of the plant, and when there are high densities, the plant just drops the leaves. So there's nothing more than just the stem. So it's a lot of production, loss of production. We also have bugs, lichens, different species that you have, but also can be a problem in vegetables and ornamentals. Tomato rust mite, it's an increasing pest species. Our topical pesticide, that's a mistake here. But you can see here the brown coloration, and here when you close up they're really tiny mites. But so far we don't have any solutions for this. We have leaf miners, and one of the increasing pest species is the leaf miner moth, Tuta absoluta. It's a big problem in southern Europe, but also it occurs also in northern Europe. And we have all kinds of caterpillars. Chrysodixis is one of the main species. So, well, biocontrol is on a pretty high level in some vegetables, but there's also a lot going on in ornamentals. And tomorrow we have this meeting, also a growers meeting in Watsonville for floriculture, and that's a big challenge. And there are several things that drive the change to biological control. With environmental pollution, emission of pesticides to the surface water. We have a lot of surface water in the Netherlands, so that's a big issue. Resistance of pests against pesticides. Sometimes growers are just not able anymore to control pests with pesticides, so they have to change to biocontrol or other options, control measures. The number of pesticides is also reduced, and when we talk about the vegetables, there is a strong demand for residue-free products. So it has to be cleaner and without pesticides. So a lot of growers now really, if you want to supply some retailers, you have to produce without insecticides. So that's a big challenge. Well, biocontrol in greenhouse crops has been applied for a long time, and there's a list of about 30 species that are released worldwide in greenhouse crops. And among them are specialists, the parasitoids, or specialist predatory mice like Phytoceras specimilis, a well-known, very important predator, specialist aphid predators, specialist entomopathogenic fungi or nematodes, or parasitoids. And then we have the generalist. So that's what I would like to talk today about. And those are predatory mites, predatory bugs, and predatory beetles. Yes, and recently, last year, there came a paper by Joop van Lenter that was telling us about the state of commercial augmentative biological control. And he mentioned that there are worldwide about 230 species used for biological control, and about 25 species of them dominate the market, about 90% of the market. And of that market, about 75% is mainly the greenhouse industry in Europe. In the Netherlands, France and Spain, those are the important countries where they use these biocontrol agents. And from those 25 species, about 6 of them are generalist predators that are widely applied. So, here's some pictures of those generalist predators. This is the predatory mouse Omniceus swisky. It's a very important one. It feeds on both whitefly. These are whitefly eggs, you can see, it's sucked empty. And this is a thrips larvae killed by swisky. This is Aureus majusculus feeding on aphids. It also feeds on thrips, on whiteflies, all kinds of pests. Really a generalist. Macrolovus pygmaeus is a myrid predatory bug, a very important one. It's similar to the vesves species you have here, or Naseniocorus used in southern Europe. Also a generalist feeding on all kinds of prey. It was mainly used in the past for whitefly control, but it also attacks spider mites and other pest species, aphids. And we have the salt-dwelling predatory mites. This is Hypoaspis feeding on the beetle larvae. It's kind of group feeding. It's very fascinating to see. So to summarize, we have different crop species, different crops, and different pest species. And you can, well, if you summarize it in one table, you can see that we have the predatory mites. This is only the generalist predators that are used. The predatory mites, phytocyte species that are leaf-dwelling, they are used in all kinds of crops. But then we have anthocorus bugs. They really need pollen often to establish, so they do very well in sweet pepper, flowering crop, but not so well in tomato. Tomato has glandular hairs, so they have problems with this. Cucumber, they don't establish. And they are trying to use these predators also in ornamentals, in roses, chrysanthemum. The myrids are mainly used in pepper, tomato, eggplant. They like the hairy plants, and they also can feed on the pollen in pepper. And then we have the salt-dwelling predators. They are mainly used in the ornamentals, because the other crops are grown in hydroponics and rock wool, so they don't establish there. So now my question, what is the role of these generalist predators in biocontrol? Why do we use them? what are the benefits and disadvantages, and what types of interactions they can be involved in. How do they act in complex food webs? So those are things I would like to discuss. Well, some benefits. I think a big benefit is the preventive releases you can do, inoculative releases. So you can start your crop with releasing these predators. The nicest example is sweet pepper, where you have young plants. As soon as they start to flower, they produce pollen and nectar, and you can release predatory mice, predatory bugs, and they can establish, even without the presence of pests. And they can build up populations to high levels. So after a while, when the pests appear, your natural enemies are already there in your crop, waiting for the pests, and then they can control them very nicely. So we call it the standing army principle. Your army of natural enemies should be ready in your crop, waiting for the pests. And they can attack multiple pest species, different pest species. That's a big advantage. You don't have to release for each pest a new natural enemy. So it can be a simple system. And you can promote them with alternative food sources. Well, but there are also some disadvantages, maybe. They can feed on other natural enemies. They are that generalist that they also attack some natural enemies that you don't want to. So we have to find out how bad that is for biocontrol. Sometimes they have a slow generation time, a slow numerical response. And if you look back to the first selection criteria that were developed for selecting natural enemies for biocontrol, they always said, well, you need natural enemies with a short generation time, so they can respond very quickly to a pest species. And that often is not the case when you use predators like predatory bugs that really need a lot of time for a new generation. And there can be some switching behavior. Because they are generalists, they can prefer maybe some other pest or food which is not the target pest you want to control. So that may be bad for control. And there's a discussion about environmental risk, the effects on non-target organisms. Well, so what I told at the beginning, we are switching a bit more from specialists. Now we had the Bacillus for spider mite control and Carcia for whitefly control, and we had this Cucumerus, which was also considered as more or less a specialist in thrips control. And we now switch to predators that control them all. And this is, for example, Amblyseius frisci, that feeds both thrips, whiteflies, and spider mites. And that's interesting because you can control them both, but you also get all kinds of interactions. And there's also theory about this, a theory by Holt about apparent competition, and it predicts that there will be a lower equilibrium density of a prey when there's a sharing predator. So one prey species has a lower density finally when there's also another prey present because of the higher predator densities. But there's also a risk, apparent mutualism. On the short term, you might have these switching behavior, and prey may escape from control because they are feeding on the other prey, and that might be worse for control. So I'll just show you now an experiment where we explore this. And this was an experiment done in greenhouses on cucumber with thrips, whiteflies, or the combination. And we both released in all greenhouse compartments this mite, Amblyseius frisci. We had separate greenhouse compartments. So you can see here, each compartment has its own door. And we had several replicates. Well, here are the results. Here I show you the population development of thrips in time. In the case when there's only thrips and the predatory mite, or thrips plus whiteflies and the predatory mite. And you see that in both cases, actually thrips gets very nicely under control. So there's no negative effect of the presence of whiteflies on the control of thrips. But if you look at the effects on whiteflies, it was really surprisingly that here you see that when there's only whitefly present, it increases. So we did not include the control without predatory mites, so there might be an effect on the whiteflies, but it's not enough. It's not able to control the whiteflies. But in a situation when there's also thrips present, you see a very nice control. So it's a very clear difference. And an explanation for this is the much higher predatory mite densities in the case when both pests are present. So here you can see this line is when both whiteflies and thrips are present, and these are the predatory mite densities at this time. So when there's only thrips or only whiteflies, these are densities per leaf, so it's still quite high. But when they are both present, you see these amazing densities of more than 100 predatory mites per leaf. So predatory mites are everywhere. And we also try to find out, well, what is the reasons for these extremely high densities of predatory mites when both pests are present? And we checked that in the laboratory when we looked at the development from egg to adult and how fast the development is and how many juveniles will die during this process, so the mortality. And you can see here that when the mixture is present, in 7 days, 25 degrees, they all reach adult stage, and there was no mortality. Whereas when there was only thrips present, we have more mortality, and there's a slower developmental rate. And this is the case with only whiteflies. We have about 40% mortality, and it's also slower. So this shows that these predatory mites, they are generalist, but also that they have a benefit by mixing their diets. They develop faster, and they control the pest better. And we did another experiment, included another pest species, which is spider mites. And also here we found the same thing. If you try to control the spider mites with only this predatory mite, it won't succeed, because it cannot enter the dense webbing of spider mites. But if you have a preventive release of these predatory mites, and they can build up populations on thrips and whiteflies, then we saw a nice control of spider mites. So this is the percentage of leaves damaged by spider mites in the case when also thrips and whiteflies are present. And this is when there are only thrips present, only whiteflies present. So here you can see actually that the more diverse the prey diet is, the more pests are present, the better the control is also of thrips and of spider mites. So these are my conclusions about these effects. So they develop better on a mixed diet, and this might be an important general principle for generalist predators. And the pest control of a specific pest species can be enhanced by the presence of another pest species when these pests share a predator. Okay. Well, another advantage I showed at the beginning is that you can promote them with alternative food sources. And we checked this for this muted predatory bug, which is very important in tomato. So we released on single plants 10, 12 couples of predatory bugs, and this is the offspring, 10 couples after 3 weeks. And you see here when you only have a tomato plant, the offspring is very low. Whereas if you add some food or prey, this is the case when whiteflies are present, they reproduce much more new predators. And so they are able to establish on plants without prey, and they can reproduce, but not so well. And these are the cases where you added an alternative food sources. The Vestia eggs, it's eggs of the floor moth. These are also used for mass producing these natural enemies, or pollen, or pred feed, it's a product, or Artemia cysts. Those are cysts of the shrimp. It's used for feeding fish, but you can also use it for feeding the predators. So we also did the next step. We went to commercial growers and applied these food sources by spraying them, blowing them with a hand blower here. And we checked how well the predators develop when you apply weekly these food sources. And here you can see, I'm not sure whether you can see it right, but here you see the red dots, which are the cysts of Artemia. And here you can see the nymphs of Macrolotus, they colored red. So they really feed on these food sources. And when we checked the increase of populations of the predators, when there's no food present, or you added Vestia eggs or Artemia cyst, you can see here that they do much better on these plots where we added the food sources. I have to say that we added 5 times more Artemia compared to Aphasia, because at the graph I showed you that Aphasia is a much better food source, but Artemia cysts are much cheaper. It's about 20 euro per kilogram, whereas Aphasia is about 800 euro per kilogram. I don't know in dollar, it's not. About 1,000, I think. So it's very expensive. And this shows that with a higher dose of a cheaper food source, and you're still much cheaper, then you had a better result in the establishment. So this is something the growers are actually applying now. Almost every tomato grower is now using this Artemia to promote the establishment of the predators at the beginning of the season. So they start planting, they release the predators, try to keep on feeding them till spring, and then they stop. And then the pests come in, and then your predators are ready to control your pests. So we also studied the possibilities for feeding predatory mites with several food sources. And here we show the oviposition rates of Amblysaius fusci. When you feed them with cattail pollen, it's Tifa latifolia, or corn pollen, or these Aphasia eggs, sterilized eggs, and Artemia cysts. And you can see that they develop very well on these food sources, but not so well on the Artemia, somehow. So it's a pity, because it's a cheap food source, but anyway, they do well on the other food sources. But, well, these predatory mites are used to control thrips. And thrips actually is a pest, but also an omnivore. It also feeds not only on the plant, but also on other food sources. They also feed on spider mite eggs, and they also feed on pollen. It's a western flower thrips. So if we add pollen to chrysanthemum leaves, or the same food sources, and we also check the oviposition rate of thrips, and then you see also an increase of oviposition rate. So these are the treatments with all the food sources, the same food sources, and this is the control with only the plant, and this is Artemia. So what you see actually is that the same food sources that do well with the predatory mites are also very suitable for the pests, for thrips. So we did not succeed in selecting a food source which is more suitable for the predator than for the pest. That was the idea, but we did not succeed. Another issue is when you provide food sources, it will also affect the predation rate of the predators on thrips. And this after 3 days, you see that when you add pollen to thrips, that the predation rate is going down. So we have a low predation rate, and we also have seen that the pest also develops on these food sources. So the big question is, what will happen if you apply these food sources in a crop? Will it promote biocontrol, or will it worsen biocontrol? Well, that's what we tested, just on simple experiments, on single plants, in cages, where we added the food sources, the thrips, the predators. And here are the results. Here you can see the development of the predatory mites, and these arrows indicate the application of food. And here you can see that, well, the predators, they establish, but not so well in chrysanthemum. Chrysanthemum is somehow they do not develop that fast as a crop like cucumber in the presence of thrips. But when you add the food source, the cattail pollen, you see that the densities go up to much higher densities. And it also goes down again, so this indicates also that the food quality is going down after a while. So you might need to apply more food. So what will happen with the thrips? Well, you might expect that the thrips is also, the food sources are also suitable for the thrips, that you have at the beginning higher thrips densities. That's what we expected. And so on the short term we also see this, but the effect is not that big. The difference is not that big. In some situations like Aphasia, we see a bit higher density of thrips compared to the control. But soon, very soon, this changes to lower thrips densities compared to the control treatment. So this indicates that there might be, on the short term, a negative effect on applying food to thrips. You might on the short term have a bit higher thrips densities, but it soon changes in lower thrips densities. And probably because there is a strong numerical response of the predators to the food sources. And they have also a relatively short generation time. Compared to other generalist predators like the bugs, they need much more time. But these predatory mice can develop, well, I showed you in the graph, from 25 degrees. Within 7 days, 7 to 10 days, they develop from egg to adult. So very soon you have a new generation and you have a strong response to the food. So my conclusion about this is, yes, we can use alternative food sources to promote predatory mice and also to enhance thrips control. And yes, we should try to select just the most suitable food source for the predator. And although these food sources are also suitable for the pest, it still enhances control. Well, these are all advantages of using generalist predators. They attack multiple pests. You have preventive releases. You can promote them with alternative food, but there are some drawbacks. A potential disadvantage is hyperpredation, and this is also an example. There are many studies done with generalist predators. Interkill predation. But in the case of interkill predation, they also always share prey, so the eventually effect on biocontrol is not always that dramatic. But in the case of hyperpredation, it's like hyperparasitism. It can really disrupt your biocontrol. And this is what we observed when we studied the biocontrol of aphids with a specialist, predatory midge, this species there. It drops eggs near aphid colonies, and from these eggs you have these larvae that feed on the aphids. But what we observed many times in the field, that some eggs were empty. They were predated, and we also observed that predatory mice were walking around there, and they turned red after feeding on these eggs. So we observed them feeding on these eggs. So we tested also in several experiments what the effect is of predatory mice on the biocontrol of thrips of aphids with this predatory midge. So we released predatory midges in a greenhouse, and we added aphids to plants, and some plants were treated with predatory mites and some not. Here you see actually a rearing sachet with predatory mites, with Swisky coming out, and you see the clear differences. On the plants without predatory mites, we had a very good control of the aphids with these predatory midges, and in this case, when the predatory mites were present, the midge was not able to control them. And we also did this in experiments. You can see it, that the densities of aphids, in the case when these mites are present, are much higher compared to when there are no aphids present, and also the number of midge larvae is much lower in the case when Swisky is present compared to the treatments without predatory mites. Something weird is going on. After a while, when you have a worse control of aphids, you get higher aphid densities, and the predatory midge is responding to that. So even when the predatory mites are present, they keep on dropping eggs near those aphid colonies. So they respond very strong to the higher aphid densities. So that's why you find higher densities of eggs of these predatory midges on the places where the predatory mites are also present. But very clearly, these predatory mites disrupt aphid control. Well, so there are several types of interactions where these predators are involved in. And one question is, well, what happens when several types of interactions occur within one food web? This is an example in sweet pepper, where we have both aphids and thrips, important pest species. So for aphids, we use these parasitoids and these predatory midges I showed you. And for thrips, we can use predatory mites and we use predatory bugs. But if we use these bugs, they both feed on thrips and aphids. And these predatory mites, well, this predator is really a generalist predator. It also feeds on parasites, aphids, or on larvae of this midge. It also feeds on the predatory mites. And what I showed, this predatory mite also feeds on the eggs of this midge. So there are already, although it's a rather simple food compared to outdoor, it's still a lot of interactions you have. Well, this is to show you that this generalist predatory bug, when you offer them both thrips and aphids, this is only thrips, predation rates, only aphids, and this is the mixed diet, that they still feed both thrips and aphids. So there's not a strong preference. So we have these interactions, and for giving advice to crows, the nice thing about greenhouses is that you actually can build your own ecosystem. You can decide which natural enemies you can release or not, and in that way you can optimize biocontrol. So here we compared two strategies. So you have to control thrips and aphids. In this case, you have to release, it's very common to release parasitoids and these predatory midges. But if you use predatory mites to control thrips, you have this negative effect on these midges, this hyperpredation. In the other case, when you use these predatory bugs, you have this intrakill predation interactions. But these predatory bugs also feed on thrips and aphids. So there are different types of interactions. And when we compare these strategies in greenhouses, separate greenhouses, and we follow the dynamics of both thrips and aphids, you can see very nicely that here is the aphid control, but we had a very good control of aphids in the strategy where we added the predatory bugs. And this is the case where you have only parasitoids and predatory midges. And this is the case where you add the predatory mites. So with the predatory mites, it goes to much higher densities. And this is the situation with thrips. Also here, we had a better control of thrips in the case when the predatory bugs were present. So this is a conclusion of these studies, is that although there are potentially negative effects of intrakill predation, these can be compensated by positive effects, like a strong effect on shared prey, apparent competition. We have this establishment prior to the pest. So intrakill predation doesn't necessarily have to be bad for biocontrol. Well, we were a bit encouraged by using generalist predators for aphid control, because so far the crows were mainly using the specialists, the parasitoids, the lacewings, the coccinellid predators, but not generalist predatory bugs. So we compared 3 species: Oryx levigatus, which is mainly used for thrips control, Oryx myusculus, what I showed you before, and there's another one, this myrid predatory bug, which was used so far mainly in tomato, but you can also release them in pepper, and that will feed in the flowers on the pollen and the nectar. You can see them here. So we compared these 3 species, and we followed the development of aphids. Well, after a while we had these results. You can already see here that in the case of levigatus, there were a lot of aphids, and with myusculus we had a few aphids, and it stayed clean when Macrolophus was present. And here are the results in the graph. Again here, levigatus, myusculus, and this is Macrolophus. These are the aphid densities in time. So we had so this is the case when you first release your predators, and then we release several times aphids, and we saw that, well, the aphids were actually not able to establish in the crop when we have these high densities of myrid predatory bugs, which is very interesting. However, thrips is the other pest species often present, and we saw that Macrolophus was not a good thrips predator. Oryx is doing much better in controlling thrips. So here we summarize the damage we measured on the fruit, and this is so the blue bars is the damage by thrips. You can see here. You can see here the damage by thrips is much higher compared to Oryx, and this is the damage by honeydew secreted by the aphids. So very little damage by aphids in the treatment with Macrolophus, but here it's very high. So next question is, well, what happens when you combine 2 species of generalist predatory bugs? We thought, well, maybe we can combine the Macrolophus for controlling thrips, aphids, with Oryx feeding on the thrips. But then you also get all kinds of interactions, intrakill predation, competition. So what will happen? So we had this food web, and now we add another predator. It's a myrid predatory bug. And we get all kinds of interactions, so it gets really complex. Well, let's first focus on the interaction between these 2 predatory bugs, the Macrolophus and Oryx, and the 2 prey species, aphids and thrips. So Macrolophus feeds on aphids. It's a good predator of aphids. It also feeds on thrips, but to a lower extent. And the opposite for Aureus. It's a good thrips predator. It also feeds on aphids, but much less. And then they both feed on the pollen and nectar in the flowers. So they also might compete for food in the flowers or for space. And there is also a negative interaction of interkill predation. So the adults of Aureus also feed on the nymphs of Macrolophus. We never observed the opposite. So by using these two predators, you get already all kind of possible interactions. And it's interesting to see how that develops in a crop. So we did these greenhouse trials in separate compartments with a sweet pepper crop. And we inoculated the crop with Aureus, Macrolophus, or both predators. And then we released thrips, followed by aphids. And we followed that for about 12 weeks. And here we see the results. This is development of aphids. And here you can see the treatment where only Aureus is present, that the aphids increase to high densities. Whereas if you have Macrolophus present in a mixture or Macrolophus alone, aphids is getting under control very nicely. And this is the other result, thrips. In the case when only Macrolophus is present, you can see here higher densities of thrips. And this is the other treatments when only is included, Aureus is included in the treatment, you have very nice control of thrips. So it seems that when you combine both thrips, both Aureus and Macrolophus, you have excellent control of both aphids and thrips, which is quite nice. Well, it's also important to know how well they establish in a crop, because they might compete. So one predator might exclude another predator after a while. And we started this, and if you look at the densities, you can see here, are the densities in the flower. Well, first it's clear that Macrolophus reaches much higher densities than Aureus, but here you can see that there is not really differences. The densities of Macrolophus are not really affected by the presence of Aureus. And also for Aureus, we found a bit lower densities of Aureus when Macrolophus was also present, but they are both established. So they seem to be okay together. So the conclusion, interkill predation by Aureus leafrats did not affect the coexistence, at least measured during this time of the experiment. We also measured in a commercial greenhouse grower for much longer, up to 8 months, and we could find them back both after 8 months. So it seems that they coexist for a while. And the combined treatment gave a better control of both thrips and aphids. Well, so we had this very good results in our experiments, but sometimes in practice it's different. What we observed is that sometimes when you have an outbreak of aphids, there's a lot of honeydew present. And these murids seem to feed also on the honeydew, and that also affects the effect on the prey. So, yeah, it's nice to work with these omnivores, but sometimes you have surprising results, because you cannot really predict sometimes how they behave. It's flexible behavior. They might switch from plant feeding to prey feeding to honeydew feeding, so it's sometimes hard to understand, but it's very interesting, and we try to understand this, how it works. So this is a short movie, but I'm not sure whether it works. Yes, it works. Yeah. So here you can see Macrolophus has been attacking an aphid, but there is a risk of attacking aphids. It's defending itself with honeydew, and it gets stick to the stylet. So after a while, it's not predating on aphids anymore, but just cleaning the stylet. The poor guy. Yeah. So these things happen as well. So to summarize about the generalist predators in greenhouse control, I think they are very important in addition to specialist natural enemies, and yeah, they deserve more attention in research. Also in our search for new natural enemies. So instead of selecting natural enemies for each pest species, I think we should much more select natural enemies that are more crop adapted and predators that prey on multiple prey. But yeah, we need to understand their behavior in realistic food webs. So greenhouse trials are really important, not only lab studies, and see what's going on in the field and how they act when there are multiple pest species present and also other natural enemies. So when selecting a natural enemy, a predator, for controlling one pest, it's important to include also other pest species that are normally present in that crop, because it can be totally different how well they control that pest species when the other pest species are also present. We saw this example with whiteflies, with spider mites. The same counts for aphids, so it's really important. So this is really an area I really would like to continue on. I'm really enthusiastic about generalist predators, and our future work is also, yeah, what I told you. It's about evaluating new species for crops, big crops, with huge challenges for ornamental crops, roses, chrysanthemum, gerbera. And also we would like to continue studying methods that enhance establishment. What kind of food can you provide, what kind of shelter or oviposition to enhance establishment. Alright, that's my talk. Thanks for your attention, and also I would like to thank my colleagues. I work together with these ladies, Ada Lehmann, Chantal Bloemaert, Renate van Holstein, and I also collaborate a lot with the University of Amsterdam, Maus Sabelis and Arne Janssen. Thank you very much.
Okay, questions for Irvin?
Yes.
You know, I've dabbled a little bit with supplementing predator mite diets with pollen. And I noticed you did some work there with pollen, with cattail and
Yes. Yeah.
foreign pollen. So, do you think differences that you observed were due to, I mean, do you know how much they ingested of the pollen? Did they just ingest different amounts? Like, is there some kind of preference for pollens? So
Yeah, there are huge differences for pollen, and also among the species of predatory mites. So one predatory mite species can do very well on a certain type of pollen, and the other not. So that's
But is it due to them having a preference for one of the pollens, or is it, I mean, they ingest the same amounts, or they just don't develop as well on one type as another one?
yeah, we don't really know. It's
Yeah, I don't either,
Yeah,
that's why I was
yeah.
asking.
yeah. I don't
What
think
about
it's
mixes of
preferences. Yeah.
What about, have you ever tried mixes of pollens at all?
No. No. It could be then. Yeah. It could be a toxic compound, or they cannot deal with, or they cannot open the pollen, or there has been some old pictures by McMurtry in the past where they show that they really grab the pollen and open it and then suck it empty. So that seems to be the method. And for some types of pollen they are not able to do that. So they cannot simply consume it. That could be a reason, or just a nutritional quality.
Yeah, that's why,
Yeah.
I don't know either, but I've seen
Yeah.
differences in different pollens.
Yeah. What's very interesting now is going on is pollen is actually commercialized now. This one company, Biological, is promoting this idea of inoculating your crop, the rose crops, with certain predatory mites, and instead of keep on releasing predatory mites, they keep on releasing, providing food. So and it works very nicely. So this is a kind of a new approach in biocontrol, much more trying to reach high densities of predators in your crop. Yes.
So like your apparent competition study that showed that two pests plus the predator were better than one, obviously that would be less expensive, self-maintaining, et cetera, et cetera.
Yeah.
But how does that go over with the growers, the idea that they have
Yeah.
to add a second pest?
Yeah, so I tell them just release some pests and then it's
Yeah.
going well.
Is that a responsibility you want?
Yeah. Now, of course, you have to take care of the risk. Although there is a method, a pest in first method. It's still used in sweet pepper. So they release spider mites and then they release the predator to establish, they try to establish a population. So the idea is not that weird, but yeah, I try to tell them, well, this pest diversity or prey diversity can be important. So sometimes, why would you, if you control whiteflies, for example, with a selected chemical, it might affect also the control of the other pests. So why not try to allow some low levels of pests when you are able to do this? It's a different story in ornamentals when the thresholds are really low for them. It's a bit harder. So in that case, it's much more safer to try to promote them with alternative food sources. But yeah, it's a different way of thinking of how to maintain these populations in your crop.
How did you feed the pollen? How did you present it so they could find it needed?
Also, they have this machine. Well, the growers, they have machines to blow it also in the crop. Yeah, it goes up to 10 meters. It's a strong wind, and then you just have very low densities, very fine, low densities of pollen. You can hardly see it, but the pollen particles are there. So that's an easy method to provide it, to bring it into your crop. Yeah. In our experiments, we just did it by hand, but in practice, you can use machines. Yeah.
Seems like the whole world is concerned about moving natural enemies around, especially generalist predators. So is it that? So one of your slides you talked about, search for new generalist predators. So what's the situation in the Netherlands? In other words, if you found a generalist, you could use it in greenhouses. There wouldn't be a regulatory concern about bringing one in, or?
Yeah, you need registration. But in the case of predatory mites, well, they don't fly, so it's also one of the criteria how well they spread, or how well they survive in cold winters. So diapausing is important. So far, it was not really an issue. I think most of the new natural enemies were allowed to use. Yeah.
One of your slides early on, you showed sort of the sterility of the greenhouses and that. I just started thinking, with the inoculative releases that you're doing and that, is there ever any concern about introducing arthropods or biomass into the food supply with the natural enemies?
Well, it can happen especially when ornamentals, when they sell flowers and there are predatory mites walking around, and then they can even be negative for people that control those. It should be clean for selling it, for exporting it. So yeah, what sometimes happens, they just kill the natural enemies at the end of the season, shortly before harvest. And another problem in tomato, for example, these myriad predatory bugs are sometimes doing so well that at a certain level they cause some damage. And so they also have to spray against the natural enemies, just to slow down to lower the densities a bit.
Yeah, the food processing itself, usually those animal parts or animals are excluded from the end product.
Yeah.
Typically though, right?
Maybe occasionally it happens that they find a predatory bug in a fruit, pepper fruit or something, but not often, no.
It sort of goes along with what Sharon was asking, the sort of counterintuitive effects of some of this.
Yeah.
Okay, Herman mentioned he and I travel down to Watsonville tomorrow for a biocontrol greenhouse floriculture conference, and it's always good for growers here to see what's going on in the Netherlands. I think maybe a shock to see what's happening, and I think that's a good thing. So let's thank Herman for a nice presentation.
And my name is Gerben Messelink. So, first of all, I work at Wageningen University and Research Center. It's a combination of applied research stations and university. I'm in the plant science group here.
Excuse me one more time. Let me just see that. Let me replace the battery. speak up. Yeah, sorry.
Yeah. Okay, so I'm working in the plant science group. Something about agriculture in the Netherlands. Horticulture is really important. I'm working in the greenhouse sector, but it's an important industry. About 70% of the production value is caused by greenhouse industry. Well, this is the Netherlands. We are a very small country in Europe. Our funding, it's mainly based, it's project-based funding. A lot of funding comes still from the government, Ministry of Economic Affairs. It's about 50%. But we are a privatized institute. And a lot of funding is coming from the Horticulture Produce Board, about 30%, and some other funding, European Community, private companies. Well, something about the greenhouses. It's mainly, well, high-tech greenhouses, glass houses we have, not so much tunnels. And they appear very sterile. If you look at this tomato production, it's all in hydroponics, not in a soil. And it looks really clean, and it is also clean. And this is an example of a nice grower who's producing sweet pepper with artificial light, so it looks really nice. But although the greenhouses are really closed and it's high-tech, somehow always pests appear, and we have mainly the common pests you probably also have, or I'm sure you also have, the whiteflies, and we have thrips coming in, one of the big problems, both in vegetables and ornamentals. We have aphids that appear.
Well, a new location, I guess, basically, where he is senior research scientist, and he does biological control, or practical biological control for the greenhouse industry, both vegetables and ornamentals in the Netherlands. He's a well-published researcher, and again, the focus is sort of understanding natural enemy biology and ecology, with the idea of making use of these natural enemies in an applied context. And he works across a broad range of different systems, and I think you'll see some of that in his presentation today. So,
yes, thank you very much, Mike, also for this invitation. It's really wonderful to be here. I hope everyone can hear me very well. Is it okay? All right. So today I would like to tell something about the role of generalist predators in biological pest control. Because in the past, I think we used to focus a bit more on the specialist natural enemies, and it has shifted a bit more to the use of generalist predators.
move that up a little higher.
Yeah.
Let's see if you can.
Is this better?
Sorry.
Not really.
Not really.
And if I speak loud, is that better?
That's better.
Yeah? I'll try to speak loud, not to shout. All right. So today I would like to tell something about well, these types of natural enemies, generalist predators in biocontrol.
Okay, it's 12:10. Time for us to get started. It's a pleasure to be able to introduce our seminar speaker for today, Herman Nesselink from the Netherlands. I think many of you have probably written or read a little bit about what has been written about Herman. He got his degree working with Joop van Lenteren at Wageningen University. And many of you may know of Joop, but basically Joop van Lenteren really advanced biological control in greenhouses around the world. And he had a tremendous stable of students that now are again around the world still advancing biological control. Herman is one of them. After his degree, he worked a little bit with a plant diagnostic firm in the Netherlands, both insects and nematodes. Kind of got bored of doing that, and they moved into a research position at Nalvik, which is an experiment station very famous for developing biological control strategies for vegetable crops in the Netherlands and around the world. And people that know anything about biocontrol in greenhouses, the name Nalvik basically comes to mind because of the tremendous advances that have been made there. There have been some major changes in the Netherlands over the years. There used to be a research station at Aalsmeer, and then one at Nalvik. Aalsmeer did floriculture, Nalvik did vegetables. They've kind of combined them. Herman has sort of moved into a new position where he is
close to the coast, but we have about 10,000 hectares of greenhouse industry, and it's mainly located in this area. It's called the Westland, and also our research station is in this area where all the industry is. It's about 50% floriculture and 50% vegetables. And there are also some other areas where they produce greenhouse industry. But also here, it was in the past more floriculture and here more vegetables. So these are the important areas. My research station is located in Blijswijk, and part of our group is working in Wageningen. And our mission is to initiate and stimulate innovations for a sustainable greenhouse sector. It's both strategic and applied research, and we have about 80 researchers all working in this greenhouse industry. In Blijswijk, it's a location where I'm working. We have about 19 greenhouse compartments where we do all kinds of experiments, crop protection laboratories, flavor, taste facilities, and we also have an innovation and demonstration center for greenhouse as energy source. So there's a lot of research going on in energy saving in greenhouses, and they want to switch from energy consuming industry to energy producing industry. So that's quite challenging. There's a lot going on, but I will not tell something about that today. It's about IPM, biological control.